Today we are going to learn about the types of culture. We are going to continue with the topics of callus culture as well as suspension culture, and one of the most important topic, that is the hairy root culture. So, in the last class we studied about the different types of cultures, like the organ cultures, where we studied about root tip culture, shoot tip culture, anther culture, flower culture and so on. We also studied in detail about the protoplast culture. So, protoplast culture is considered to be a game changer in the plant tissue culture. However, if we talk about the medicinal plants, generally the best you can say breakthrough which has been found, that is the hairy root culture. So, hairy root culture is a culture where we can increase the type of the secondary metabolite product secondary metabolites in the plants by adding the desired message in the DNA. Okay. So, here we can manipulate the DNA of the plant using a specific bacterium and then do the plant tissue culture, leading to the production of the desired secondary metabolites. So, it is one of the best breakthroughs for especially those people who are working in the area of medicinal plants. So, the name hairy root culture was mentioned in the literature by Stewart et al. 1900. A large number of small fine hairy roots covered with root hairs originate directly from the explant in response to the bacteria named Agrobacterium rhizogenus, and this infection, it is termed as the hairy root culture. So, this finding was done, basically what happened, that the scientist, he had gone he was just checking few plants, and he found that a plant had developed some kind of infection. And after that what he did that from those infected cells, he took few cells and he started studying those cells. He found that the bacteria had invaded the plant cells and was able to send its message into the plant cell using the specific you can say type of plasmid which it contains, which is known as the RI plasmid. So, this led to his curiousness, and he performed the entire study and tried to imbibe few messages into the RI plasmid of the bacterium and tried to transfer that message into the plant cell, and he was successful in that. So, this is how the hairy root culture was initiated. Agrobacterium, it basically recognizes small signals exuded by the wounded plant cell and becomes attached to it. So, if any kind of wound is there on a plant, Agrobacterium can easily find that where this is there, where this wound is there. It reaches to that wound, starts growing on that particular wound, and gradually transfers its message into the particular plant cells using its RI plasmid. So, this RI plasmid, the full form for RI plasmid is root inducing plasmid. The bacteria genetically transfers part of the RI plasmid called the transfer DNA, that is tDNA, to the plant genome where it gets expressed. So, this is the structure of the RI plasmid. So, here you have different regions in this RI plasmid. Basically, I will first discuss the three other regions. One is the virulence regions. This region is responsible for the infection. It helps in developing such proteins, will help in dividing the cells and transferring it to the areas wherever the wound is there. So, it is responsible for the virulence of the bacteria. This is the origin of replication. If the cell has to start dividing, the origin part message is there in this part of the RI plasmid. Then opine catabolism, like if any kind of the metabolic processes performed by the particular agrobacterium, what are the enzymes required, what are the processes involved, that is governed by the opine catabolism part. Now, we move further, this is the conjugative transfer part, which helps in transfer of the RI plasmid into the plant cell. And here this yellow region which you can see, this is the T region, which is known as the transfer DNA. This region is going to get transferred inside the plant cell. So, this region has different regions. This is the left TNA border, this is the right tDNA border, and in between we have area. This purple color part is the auxin production area, it is depicted like this. Then you have the cytokinin production area, this is the opine synthesis area. So, you have different areas in this particular whole strand of the DNA. I should not call it DNA, it is a RNA, which is having the entire message of the auxins, what amount of auxins have to be produced, what amount of cytokinins have been produced, what amount of other secondary metabolites have to be produced. This whole message is there in this tDNA of this RI plasmid. Okay. Now, what happens is that if we take, this is the bacterial cell and this is the plant cell. Now, in this bacterial cell you have the RI plasmid and this is your plant cell which is normally functioning. What happens is that through this origin, through this particular origin of replication as well as through this conjugative transfer part, this particular bacteria, it creates a pathway between the plant cell and the bacterium. This is Agrobacterium rhizogenus, this is our plant cell. So, this RI plasmid, it will generally pass inside the cell of the plant and these are the chromosomes of the plant inside the nucleus. This particular message is then transferred. If I talk about, this is the chromosome of the plant cell, this is the RI plasmid, it is going to reach to this plant cell, shift its or transfer its tDNA into the plant cell and then it is going to leave. So, due to the infection, what has happened? The entire DNA fragment, RNA fragment has been replaced from the bacteria into the plant cell. Now, the chromosome of the plant cell has the entire message that how much auxins, how much cytokinins, how much secondary metabolites have to be produced and they start dividing, they form a callus and from the callus generally hairy roots are developed, which is why the name has been given hairy root culture, and they are generally very good or a very large scale producer of secondary metabolites. A compound named chiconin, which is a dye, it is manufactured by a Japanese company, chiconin. It has been manufactured till date by the procedure of hairy root culture. So, this is one of the most, you can say, widely known application of hairy root culture. So, explants are wounded and then inoculated with Agrobacterium rhizogenus. Usually 2 to 3 days later, the explant can be transferred into the solid medium with antibiotics to kill or eliminate reductant bacteria. The hairy roots will be induced within a short period of time, which varies from a 1 week to over a month depending upon different plant species. The decontaminated roots, hairy roots can be subcultured on phytohormone free medium. Proliferate by increasing the rate of cell division and cell elongation to produce hairy roots. So, we can make different changes inside the particular medium depending upon which we can either increase the rate of cell division, we can decrease the rate of cell division, we can increase the cell elongation. And from the hairy roots itself, we can either develop the callus or we can develop the entire plant also. And many a times from the hairy roots only the secondary metabolites can also be isolated. Now, we come to the different types of culture which is based on the types of the medium. Till now we were somewhere we have discussed about based on the type of the plant part which we had used. We also discussed the specialized techniques like the protoplast culture as well as the hairy root culture. Now, further we will be discussing types of culture based on which type of medium we are going to use. So, first of all there are two types of culture, one is the suspension culture or the Callus culture, which is also known as the static culture. So, what is the basic difference here? What we did, we have taken the explant and we have dipped the explant in a medium where the explant grows in an uncontrolled way and forms a callus. Now, if we put this callus in a liquid medium and we do agitation, the callus cells will break down and they will give us the dispersed cells. So, here in this medium what is there? The nutrient medium is completely static in nature, it is not moving, while in suspension culture it is completely agitating, means completely moving. So, it is one and the same thing, but the difference is that in suspension culture the medium is liquid in nature and it keeps on moving, shaking, while in Callus culture, in the static culture, the medium is solid and it does not move, it does not agitate. So, that is the static medium. So, the static one is known as Callus culture, while the moving one is known as the suspension culture. The content which is present inside is the same. Further, suspension culture are of different types. First, we will discuss about the suspension culture. It involves the active proliferation of callus as undivided units suspended or submerged in a liquid medium. So, you have the callus which is divided into or suspended into a liquid medium basically. Okay. The nutrient medium in this case is in constant agitation, it keeps on agitating so on to prevent the cells from settling or aggregating into form a lump. So, it has to be moved completely to avoid any kind of settling down, sedimentation or clumping together. Suspension cultures are normally initiated by transferring pieces of undifferentiated callus to a liquid medium which is agitated during incubation. So, how we are going to prepare the suspension culture? We will take few callus cells which are undifferentiated and then we will put it in a liquid medium and keep on agitating it. Now, there are different types of suspension culture and I will come back to this picture. Here you can see that the suspension culture of three types: batch culture, semi-continuous culture and continuous culture. See, before we move further, we are going to discuss later how the growth and maintenance of the cultures is done. So, this is in brief related to that also. So, if you do any kind of culturing like you have done a callus culture. Now, after a period of 3 to 4 weeks, what will happen? The static medium is going to give all its nutrients to the callus. So, after that what will be there? There will be no nutrition inside the medium. So, the callus will slowly die. Okay. So, to avoid that, the other thing which is very much important in plant tissue culture is subculturing. You have to do the subculturing, and for subculturing one of the best technique is, rather than going for callus culture, we should go for suspension culture. Okay. So, in suspension culture you can say I can either quote it like that suspension culture is a technique to perform the subculturing of different plant tissue cultures, so that the the process of plant tissue culture is continuous and it is sustained. Okay. So, here you have the batch culture, semi-continuous culture and continuous culture. In batch culture, what happens is that you have taken a fresh medium, you have placed a fresh callus, you have agitated it continuously, and after a period of say about 3 to 4 weeks, you removed the cells from the medium, transferred it into a fresh batch of culture, and then again the same process of agitation went on. So, what is happening is every time, like every 4 to 6 weeks, you are replacing fresh medium, you are taking the cells and putting it into a fresh medium. So, batch to batch, like you have the first batch of 6 weeks, then you go for the second batch of 6 weeks, then next 6 weeks. This goes on one by one. This is the batch culture. After that, the second type is the semi-continuous culture. Now, in semi-continuous culture, what is done is that at particular point of time, you have a inlet and a outlet. You allow some amount of the fresh culture coming in, some amount of used up culture going out. Then again you wait for a particular time, you stop this procedure. After say 15 days, again what you did, you entered some amount of fresh culture, removed some amount of used up culture. So, this is a semi continuous culture where at a particular time interval you are putting in fresh culture medium, so that the plant is the cells do not feel short of any kind of nutrients. Okay. So, this is semi continuous culture. And last is the continuous culture. In continuous culture, what happens is that in the compartment where the suspension culture is there, you have a continuous inflow of the nutrient medium and continuous removal of the used up medium. So, that is why it is known as a continuous culture. Again, continuous culture is of two type, open type and closed type. Now, what is a open type? In open type of culture, what happens is that you basically you monitor that or regulate that how is the use you can say utility or utilization of the all the chemical constituents taking place. Means in a particular system you have a nutrients. Okay. So, these nutrients are getting used up by the plants, the callus cells. So, you keep on monitoring or regulating that okay, now 50% of the glucose has been utilized. So, I have to add more 50% of the glucose. I have to check that whether it is having any shortage of vitamins. Yes, if it is short, I will add that amount of vitamins. And this analysis is done by two techniques or the regulators or the readings, they are taken either by the turbidostatic method or by the chemostatic method. Okay. So, what is turbidostatic method? In turbidostatic method, what is done that the sample of the callus, it is checked for turbidity, amount of turbidity. If the turbidity is increased, it means there is lot of waste material, plant waste is increasing in the medium and we need to replace it with a fresh medium. So, that is how you check the turbidity. So, there are specialized turbidometers which are there. And nowadays, we can use various softwares where the continuous reading can be taken and we can get the information that yes, the turbidity has increased. So, the regulator will work itself, it will start input or it will start the intake of the particular fresh nutrient medium. Okay. Now, it feels that the turbidity is sufficient. So, what it will do? It will lower down the rate of entry of the particular fresh medium. So, it will now slow down. Now, after some time again the turbidity increased. So, it will increase the rate of inflow of the fresh medium. The second technique is the chemostatic technique. where for example, nowadays it is very easy to measure the blood sugar. Same way we can easily measure the sugar level in the particular samples. So, we can keep on analyzing sugars, proteins, vitamins, minerals or any other compounds which we have put inside the sample, and the results, depending upon the results of the analysis, we will decide that at which rate the inflow of the fresh medium has to be taken place and at what rate it has to be removed from the system. So, that is why, because a continuous flow is going on, this type of system is known as a open type of continuous culture. The second type, that is the closed type of continuous culture, in this what is there, that it is a, you can say, fixed system, where at a particular rate you have decided that at 1 ml per minute fresh medium will be coming and used up medium will be leaving the chamber. So, this is a simple closed type of system. Here no monitoring or no regulation is done, and this is also a type of a continuous culture. So, this is, I will say, closed system is a old method compared to the open system. The best method, or the most, you can say, giving us the best produce yielding, is the open type of continuous suspension culture. The second one is the Callus culture. So, Callus culture, where you have a mass of undifferentiated plants cells growing on solid medium from the plant part or the explant. It may initiate from explants of any multicellular plant. So, you can see these are very bright colored green material looking like a amorphous material which are differentiated from a normal cell, and further they will again re-differentiate into a new plant. So, we are coming back to the batch suspension culture. As I told you, it means the culture in a fixed volume of culture medium. In general, a nutrient medium and cellular inoculum are mixed, aerated and allowed to grow. In batch cultures, as the cell grows, the medium is depleted of the nutrients and metabolic byproducts from the cell accumulate. The system is closed with respect to the addition or removal of culture except for circulation of air. Then we discussed about the semi-continuous culture. The system is open here. There is a periodic removal of the culture and addition of fresh medium, due to which the growth of the culture is continuously maintained. Continuous suspension culture, this system is open here also. If in this system the volume of the culture remains constant and fresh medium is added continuously to fixed volume of the growing culture and withdrawn. It is again of two types, one is the open type and another is the closed type. In open type we have two types, one is the chemostat, where we can continuously check that what are the different chemicals present in the particular sample, and depending upon that we can regulate new medium and balance the harvest of equal volume of the culture. It is useful to study the steady states of the plants. In turbidostat method, generally the cell density is set at the predetermined level and it is monitored using the optical properties of the culture, and new medium is added to maintain the cell density within limits. Particularly valuable for work at low cell densities and offers a simplified system for study of effects of physical factors and growth regulating substance in growth rate and associated metabolism. Then last is the closed type of suspension culture. The used medium is replaced with fresh medium and hence the cells from the used medium are mechanically separated and added back to the culture, and thus the cell biomass keeps increasing. So, here in this particular lecture we have studied about what are the different types of hairy root cultures, what is suspension culture and its different types, and then Callus culture. We have to note that suspension culture and Callus culture are the best techniques for performing subculturing of our plant tissue culture. Thank you very much.